Good morning, good afternoon, and good evening, everyone. And thank you for joining us for our webinar titled Remodeling of Pancreatic Innervation in Diabetes. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This program was developed through a collaboration between the American Physiological Society and Miltenyi Biotech, so a big thanks to them for making this program possible. Today, we're very pleased to be joined by Dr. Sarah Stanley and Dr. Alexandra Alvarsson, respectively Assistant Professor and Senior Scientist at the Diabetes, Obesity, and Metabolism Institute at the Icahn School of Medicine at Mount Sinai. Doctors Sarah Stanley and Alexandra Alvarsson will be discussing the use of whole organ imaging of the pancreas to reveal close interactions between nerves and islets and dynamic regulation of islet innervation in diabetes. And with that, I'm very pleased to welcome Dr. Sarah Stanley. Sarah, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks, Liam. Thank you so much for the introduction, and thank you all for being here. We're really excited to tell you about our work on pancreatic innovation in diabetes. I'm going to start by giving an overview of what we know about neural control of glucose metabolism, and specifically about pancreatic innovation, and then Alexandra will tell you more about the work we've been doing. Disclosures. So for most species and most of human history, food supply has actually been pretty unpredictable. Although there are periods when food is abundant, there are also long periods where it's scarce or even absent. Yet despite this, blood glucose is maintained within a pretty narrow range between 70 and 140 milligrams per deciliter. And one reason why this is important is because glucose is virtually the sole fuel for the brain. Although the brain only weighs 10% of body weight, it consumes between 60 and 70% of whole body glucose in the resting state. And actually the system for providing glucose to neurons is really highly evolved and beautifully designed. So glucose in the circulation is taken up into the extracellular space around neurons through a very high affinity glucose transporter, GLUT1. Glucose then goes into the neuron itself through an even higher affinity transporter, GLUT3. And there it's trapped within the neuron by phosphorylation to glucose 6-phosphate using hexokinase 1. So this system means that under most circumstances, neurons can maintain their supply of glucose. However, they do rely on maintaining blood levels of glucose. And if blood levels are very high for a long period of time, this leads to high extracellular glucose in the CNS and can have quite severe osmotic effects, which eventually lead to loss of consciousness and even death. Conversely, hypoglycemia in the circulation rapidly leads to a loss of fuel for the CNS and is, again, very dangerous, leading to loss of consciousness and even death. So there are multiple systems that have evolved to try and maintain blood glucose within a narrow range. And we know that some of these systems involve the CNS and peripheral nervous system. So in order to do so, there need to be mechanisms whereby the brain can sense blood glucose and also have effects on peripheral organs to return it to normal. So we know that there are specialized neurons that use glucose not only as a fuel but also as a signal. And these neurons are actually pretty similar to the endocrine cells within the pancreas. Some of them are beta cell-like in that they're activated when glucose levels are high. These are called glucose-excited neurons. Others are more like alpha cells, and these are switched on when glucose levels are low and are glucose-inhibited neurons. And when we look at the location of the glucose-sensing neurons, we can see that they're actually distributed through the CNS to form a network. They're particularly found in the hypothalamus and also in the brainstem. And this figure summarizes many decades of work looking at electrophysiological studies in ex vivo slices and calcium imaging. And as you can see, between 5 and 10% of neurons in the hypothalamus are either glucose-excited, GE, or glucose-inhibited, GI. And just like the pancreas, there isn't an anatomical segregation between these populations. They're intermingled within the hypothalamic nuclei. We do know the connections of some of the glucose-sensing neurons, but many of them we don't. But we know that the areas where they're found are connected to the autonomic outflow tract. So, for example, the paraventricular hypothalamus, the PVH, is connected to the intermediolateral nucleus in the spinal cord, which regulates sympathetic outflow. And areas such as the lateral hypothalamus are connected to the dorsal motor nucleus of the vagus in the brainstem, and these regulate parasympathetic outflow. There have actually been many studies over the last 50 to 60 years which have looked at the role of the autonomic nervous system in glucose regulation. These studies include nerve transection studies, electrical stimulation of nerves, the use of agonists and antagonists. And to summarize these, the parasympathetic nervous system, which is generally active in the fed state, increases insulin release. Conversely, the sympathetic nervous system, which is generally active during the fasting state or even hypoglycemia, acts to increase glucagon and suppress insulin, and also regulates insulin sensitivity and glucose output from the liver. So most of these studies have been performed in animal models, but there's also a significant amount of evidence to show that neural regulation is important in humans as well. And this comes from rare pathologies and also from clinical studies. So we know, for example, that individuals who have hypothalamic pathology, such as space-occupying lesions or inflammation, have high insulin levels and insulin resistance, and may even have impaired glucose tolerance. We also know that there are abnormalities in individuals who have demyelination, for example, due to multiple sclerosis. And finally, in individuals who have autonomic neuropathy, there is evidence of impaired hypoglycemic responses, particularly in the production of glucagon. There have been many clinical studies in humans as well, and this just illustrates a few of the important ones. So, for example, functional MRI studies have shown that there is regional CNS activation with hypoglycemia, suggesting that there are mechanisms by which human brain can sense changes in blood glucose. There's also pretty strong evidence for something known as the cephalic phase insulin release. So this is a reflex when food is placed in the mouth, and before blood glucose is actually changed, a neural arc triggers the release of insulin to prepare the body for the coming food. Studies have also used deep brain stimulation or transcranial magnetic stimulation in individuals who have Parkinson's disease or diabetes or depression, respectively, and have shown that glucose metabolism is altered, in particular a change in hepatic glucose production with deep brain stimulation. There have been several studies that have looked at the effects of vagotomy in individuals and their effect on glucose tolerance. And although these are not conclusive, there are at least some which suggest that glucose tolerance is abnormal in individuals with vagotomy, and if anything, it's impaired. And finally, we know that in individuals who've had pancreatic transplants and before there's re-innovation, there is an abnormal response to exercise and hypoglycemia. So what these studies suggest is that neural regulation of glucose is important in animals and in humans, and it's possible that we might be able to harness these neural signals to control blood glucose. But in order to do so, we need to have a better understanding of the anatomy and function of these nerves. So what about the pancreas? Well, this is actually innervated by several autonomic systems and also sensory systems, which I'll now go through in more detail. So the parasympathetic inputs into the pancreas arise from the dorsal motor nucleus of the vagus in the brainstem. And anterograde tracing studies from this region has shown that they provide inputs into the intrapancreatic ganglia. There have also been some functional studies which show that if you activate the dorsal motor nucleus of the vagus, then you can see CFOS, which is a marker of neuronal activation, in about 10 to 30% of the intrapancreatic ganglia. Another input into the pancreas is actually from the sympathetic nervous system. So here the preganglionic neurons are in the intermediolateral column of the spinal cord and project to the celiac ganglia in the abdomen via the splanchnic nerves. The sympathetic postganglionic fibers then project from the celiac ganglia to the pancreas and have been shown to innervate the intrapancreatic ganglia, the islets, the vasculature, and also lymph nodes in and around the pancreas. There's extensive sensory innervation of the pancreas, and this has been shown by beautiful studies by Fasanella et al. Some of the sensory innervation is vagal, and this involves both the exocrine and endocrine pancreas. And lovely work from the Caicedo group has shown that these neurons are primarily chemosensors. From Fasanella et al., we know that there's also extensive spinal sensory innervation, involving T5 through T13, and these spinal sensory nerves express substance P, TRPV1, and CGRP. And it's likely that these sensory neurons from the spinal cord are both mechano- and chemosensors. In addition to these, there's also an extensive network of neurons and intrapancreatic ganglia that form a mesh within the pancreas. The intrapancreatic ganglia are often adjacent to the islets, and studies from Tang et al. Have shown beautifully that these form neuroinsular complexes. Both the ganglia and the fibers from the ganglia are primarily cholinergic, but they also express neuropeptides such as vasoactive intestinal peptide. There's been really extensive examination of pancreatic innervation in 2 dimensions, and these have been really beautiful studies which have shown both the connections between the nerves and also between individual endocrine cells within the islets themselves. And I've just pointed out a few of the most beautiful studies there. What these studies have shown is there's actually extensive parasympathetic and sympathetic innervation in rodent islets. But if you look at human islets, then the innervation seems to be much more sparse. And rather than being parasympathetic, it seems to be primarily sympathetic. And this is work from the Caicedo group again. So the advantages of 2-dimensional imaging are that it allows really high-resolution imaging, particularly of the fine structure of the innervation within the islets. And as I mentioned previously, you can identify the nerve fibers and how they are adjacent to individual endocrine cells. However, the downside of the 2-dimensional imaging is that the pancreas itself is very heterogeneous. And in order to process a whole pancreas, particularly a human pancreas, this requires very laborious serial sectioning. The other disadvantage is that if you're looking at filamentous structures, particularly nerves or vasculature, then tracing them over long distances can be quite difficult. And because you're taking snapshots through the pancreas, you may miss regional differences across the whole pancreas. And the panels here in the top show some 2D imaging that we've done, showing insulin in beta cells and tyrosine hydroxylase, TH, and synapsin using cryosections. Whereas the lower panel is 3D imaging in a small piece of pancreatic tissue stained for insulin and the neuronal marker synapsin. So what's happened recently is that there's been a resurgence in optical clearing methods to examine pancreatic structure. And in the resources and here in this slide, there are some really extensive and beautiful reviews that have talked about the different clearing methods involved. So we've used iDisco Plus, which Alexandra will describe shortly, but also fructose clearing and ECI clearing. And I'm just giving you a snapshot of some of the images that we obtain with these. Alongside the resurgence in optical clearing methods, there's also been advances in volumetric imaging, particularly like sheet microscopy, optical projection tomography, and scape microscopy. And the third component has also been the progress that's been made in the software for 3D volumetric processing. These include open source such as ImageJ, but also commercial software such as Neuralucida and Imaris. There's been quite a lot of work done recently looking at pancreatic innervation in 3 dimensions. I'm just going to highlight a few of these studies. There's also been a lot of work done looking at the islet structure and distribution, which I haven't got time to discuss here, but if you want to look further, then the work from the Hara lab is excellent. So the first paper I'm going to talk about briefly is a study from Chow et al. From 2012, which examined sympathetic innervation in diabetic mice. Here they used fluorescent lectins and immunolabel to examine vasculature and innervation, and a commercial tissue clearing focus clear, followed by confocal imaging. We can see in their paper that they show that there's intra-islet sympathetic fibers which are particularly associated with the vasculature, and that this is increased in SCZ mouse models of diabetes, but not in non-obese diabetic mice. And the illustration here is work that we've done to show that the TH, the sympathetic fibers, do indeed follow vasculature in the pancreas. There have also been studies looking at innervation in intrapancreatic ganglia in a genetic model of diabetes, the DB-DB mice. Again, using immunolabel to look at innervation and islets, and a commercial solution Rapiclear followed by tiled confocal imaging. And this study really beautifully illustrated the structure of the intrapancreatic ganglia, and also that as these ganglia increase in size, then their connections to surrounding islets increase in number. And they also demonstrated that pancreatic sympathetic innervation is increased in the DB mice. And more recently, there have been several papers that have looked at human pancreatic tissue. So from Tang et al., there was an examination of the sympathetic and parasympathetic innervation in human pancreas. And in stark contrast to the 2D imaging, they suggest that there is some sympathetic innervation of the vasculature and also the islet core itself. And they also demonstrated parasympathetic innervation of the islet core. Chen et al. Looked at sensory innervation of human islets and intrapancreatic ganglia and showed that the sensory fibers go to the ganglia themselves, but not the islets in human tissue. And finally, lovely work recently from Martha Campbell-Thompson's group has shown that there's sympathetic innervation of islets and that this is actually reduced in autoantibody positive individuals compared to either controls or those with type 1 diabetes. So with that, I'm going to hand over to Alexandra, who's going to tell you more about our work.
Thank you, Sarah. So the aims of the study that I will talk to you about today was to use optical clearing by Iriscope Plus combined with advanced 3D rendering to determine the distribution of islets and innervation throughout the whole pancreas in healthy animals, in mouse models of diabetes, and in human donors without and with diabetes. More specifically, the aim was to compare the duodenal versus the splenic region, and we did this in C57 black 6 mice, adult male mice. We also wanted to compare the non-diabetic versus diabetic state, and to do this, we used non-obese diabetic or NOD mice. These mice were females, 12 to 16 weeks of age, and this model spontaneously develops type 1 diabetes with insulitis or islet inflammation. These mice were defined as diabetic by 2 consecutive blood glucose measurements above 300 milligrams per deciliter. And we used non-diabetic littermates with blood glucose below 200 milligrams per deciliter as non-diabetic controls. The average non-fasting blood glucose in these mice was 115 in the non-diabetic group and 495 in the diabetic group. To study the more progressive effects of diabetes, we used the multiple low-dose streptozotocin or STZ-induced mouse model. These mice were again C57 black 6 males, 10 weeks of age. This is a progressive model of type 1 diabetes. Streptozotocin is a beta cell toxin that after multiple low doses induces insulitis or islet inflammation. These mice were treated with 5 low doses of STZ, and then they were sacrificed 5 days or 15 days following the final STZ injection. We used non-STZ treated littermates as controls. The average non-fasting blood glucose in these mice was 123 milligrams per deciliter in the control group, 259 milligrams per deciliter on day 5, and 430 milligrams per deciliter on day 15. Finally, we managed to acquire samples from human donors without and with type 2 diabetes. So first, a few words on the methods, beginning with the tissue harvesting. So we used non-diabetic and diabetic mice, which were perfused with heparinized saline, followed by 4% paraformaldehyde, which was kept on ice. Tissue was then, pancreata were then dissected, and the surrounding organs and the adipose tissue was removed. And finally, the tissue was dissected into the splenic and the duodenal region for anatomical comparisons. We used optical clearing by Iriscope Plus, and we used a protocol adapted from the work of Nicolas Renier and colleagues in 2016. This is a fairly long protocol, beginning with tissue pre-treatment. This is done to remove soluble and hydrophobic lipids, and then tissue is treated with peroxide to reduce tissue autofluorescence. During the immunolabeling step, pancreata were stained for insulin to detect beta cells, for glucagon to detect alpha cells, for NF200, which we use as our paneuronal marker, for TH, which we used as a sympathetic marker, and VACHT, which we used as our parasympathetic marker. During the antibody incubation steps, we used a modified PTXWH buffer to improve neuronal labeling of pancreatic tissue. And this was one of the adaptations that we did from the original protocol by Nicolas Renier and colleagues in 2016. And we have written up a bioprotocol which will be out any day soon, where we really go into the details of this modified protocol. Finally, we perform the tissue clearing step, where the samples are immersed in the benzyl ether, or DBE, which has a refractive index of 1.562. The samples are stored and imaged in DBE. And this brings us to the imaging and analysis part. So here you can see a pancreas embedded in Agarose before the tissue clearing protocol and after the completion of the protocol. And as you can see at the end, the tissue is rendered optically transparent. So we use this transparent tissue to image at a light sheet microscope. And the advantage of using a light sheet microscope is that you illuminate a very thin slice of the sample at once, and you can actually illuminate the entire sample. So you can very rapidly acquire really large Z-stacks. We imaged the samples at 1.3x, 4x, and 12x magnification, and then some of the samples were also imaged using a confocal microscope at 10x magnification. I will talk more about that later on. Finally, all the Z-stacks that we acquired were processed in Imaris to render 3D images, to perform quantifications, volumetric analysis, and distance analysis. So some considerations about the imaging. We used light sheet data acquired at 1.3x magnification to image entire hemipancreata. This we used specifically for islet distribution analysis. We used light sheet data acquired at 4x magnification to analyze innervation. And we used confocal data acquired at 10x magnification to image ganglia, alpha cells, and beta cells, because with this analysis, we needed to be able to distinguish individual cells. Here is, basically this is the Z-stack that we get from the light sheet microscope that we acquire via imaging, and this is the 3D reconstruction that we do in Imaris. Here you can see insulin in green and NF200 in magenta. Again, we use this only for the islet distribution across the pancreas. In this case, it's the splenic region of a mouse pancreas. So moving back to the animal models. So again, we use C57-6 black mice to look for the regional variation in islet distribution. And then we use non-obese diabetic or NOD mice, which spontaneously develop type 1 diabetes and insulitis, to compare the non-diabetic versus the diabetic state. And then we used STZ mice, streptozotocin-induced mice, to monitor or to address the progressive pathology of diabetes. In C57-6 black mice, we found regional variation in the islet volume and in the insulin content between the duodenal and the splenic region. We found a significantly higher islet volume in the splenic versus duodenal region, and we found a significantly lower insulin content in the duodenal versus a lower insulin content in the splenic versus the duodenal region. And this is similar to previous reports on regional variation in the pancreas. So this is a nice validation of the methodology. We then looked at NOD mice, and we found that there was a dramatic loss of islet volume and islet density in the diabetic state of these mice, with a slightly greater effect in the splenic region of the pancreas. In the STZ model, we found that low-dose SSE treatment progressively reduces islet volume and the insulin content, but not the islet density. And the reason why we didn't see an effect on islet density is really because in this model, it appears as if the islets become fragmented. rather than them just losing entire islets. And in both these models, these pathological features correlated with increasing blood glucose. In human donors, again, these donors suffered from type 2 diabetes, the diabetic ones, and then we had samples from non-diabetic donors. And you can see the 8 samples that we have side by side. C1 to C5 are the non-diabetic donors, and D1 to D3 are the diabetic donors. And as you can probably see from these images, the beta cell volume and the islet numbers and also the innervation was highly variable in these samples, even though they were all sampled from the same region of the pancreas. So the tail region towards the middle of the pancreas. And this is of course expected with humans. But on average, we could see that the islet volume was lower in diabetic donors compared to non-diabetic donors. We also found that the islet number per cubic millimeter was greater in humans than in mice, whereas the beta cell volume remained similar. We then moved on to look at the islet volume distribution. And on the right here, I'm showing you a video that sort of illustrates how we did this analysis. We used 1.3x data, but this model is actually from 4x data just for illustrative purposes. So here we used Imaris to determine the volume of the islets, and these islets are colored with an overlay based on islet volume. So the really large islets are shown in magenta, and the really small ones are shown in cyan. And on the left, this is a graphic representation of this. So small islets consisting of 8 or fewer beta cells, so those are sort of proto-islets, are shown in light blue, and really large islets with volumes above 500,000 micrometers, cubic micrometers, are shown in black. And as you can see, there's not much regional variation in the islet volume distribution in control mice. But when looking at the diabetic models, the NOD mice in particular, we saw a dramatic shift towards smaller islets in the diabetic state. In STZ mice, we did not see a similar shift, which suggests that islets are affected sort of to the same degree by diabetes. But when looking at human donors, we again saw a very dramatic shift towards smaller islets in humans with type 2 diabetes. We then moved on to look at glucagon as a measure of alpha cells. And on the right is a non-diabetic NOD mouse, and on the left is a non-diabetic NOD mouse, and on the right is a diabetic NOD mouse. Glucagon is labeled in blue, insulin in green, and NF200 or panorama marker in magenta. And as you can see, there's a dramatic loss of insulin in the diabetic mouse pancreas. In NOD mice, as expected, we saw a dramatically increased glucagon to insulin ratio in the diabetic state. In the STZ model, we again saw a reduced glucagon to insulin ratio as expected. It was not as dramatic as the NOD mice. What is interesting to note here is that the STZ model actually had a reduction in alpha cells 15 days after the STZ treatment. We then moved on to look at the endocrine innovation. And as you can see, islets are densely innervated. And we defined any innovation that made contact with insulin-positive beta cells as endocrine innovation. We also measured this based on glucagon-positive alpha cells. So we used 2 different measurements, but we only used alpha cells for the diabetic models. But basically any innovation that goes into the islets, that makes contacts with the islets. And on the right, you can see this innovation highlighted in white and now in yellow. So these are volumes that we created in Imaris around this innovation to pull out the volume. And when looking at control mice, we saw a quite dramatic regional variation in the endocrine innovation, with more endocrine innovation in the duodenal versus the splenic part of the pancreas. And we saw this both when calculating this as the total nerve volume per islets in cubic micrometers on the left, and when calculating it as a percentage of the islet volume, so a percentage in this case of the insulin volume. Interestingly, when we compared the endocrine innovation with the exocrine innovation, we found that endocrine innovation was dramatically enriched in mouse pancreata. It's about 10 times enriched. And this is very interesting based on the fact that endocrine cells only make up a few percentages of the total pancreatic volume, but still they receive so much more innovation. In the NOD model, we found that islet innovation was increased, particularly in the splenic pancreas, when we calculated it as a percentage of the insulin volume. These mice, however, as I pointed out before, have a very dramatic loss of insulin. But when looking at endocrine innovation as a percentage of the glucagon volume, we didn't see any change. So this suggests that endocrine innovation is preserved in this model in diabetic state, or even slightly enriched. In the STZ model, we found increased endocrine innovation again. And when looking at endocrine innovation as a percentage of the glucagon volume, we saw a very strong enrichment actually at day 15 after the STZ treatment. So this again suggests that NF200 positive endocrine innovation is preserved or even enriched in the diabetic state of these mice. Moving on to the humans, and again, here we can clearly see that the islet innovation is quite variable in the humans, but if anything, it's greater in samples from diabetic individuals. Interestingly, when looking at the exocrine innovation and comparing that to the endocrine innovation, there was no enrichment in the exocrine innovation versus the endocrine innovation in humans. So this suggests a species difference, and it's actually in line with several previous reports that have shown that human islets are just not as densely innervated as mouse islets, although there is conflicting data when it comes to that, but at least with NF200, using this methodology, there was no enrichment. We then wanted to look more closely at the interactions between the nervous system and the islets. So here's a pancreas with insulin in green, NF200 in white, and we performed a distance transformation analysis shown here, where you calculate the distance between islets and innervation, and purple islets here are touching the nerves, and dark blue islets are very close to the nerves. So we use this distance transformation to calculate how closely situated islets are to pancreatic nerves. So that's what it looks like. And we pulled out islets that were situated less than 1.6 micrometers from the innervation. In control mice, we saw that about 6% of islets were closely situated less than 1.6 micrometers from NF200 positive innervation. And it didn't really vary depending on what region we looked like. It remained about 6%. In NOD mice, we found that more islets were situated closely to nerves. And in non-diabetic mice, this was 15%, diabetic mice 10%, so slightly more. And I should mention here that non-diabetic NOD mice can actually be considered pre-diabetic, since they will spontaneously develop diabetes at some point, most of them. When looking at the glucagon, however, we define this as alpha cell clusters. We saw that in the diabetic state, more alpha cell clusters were located in a close proximity to the nerves. In the non-diabetic state, it was again 15%, so very similar to that of insulin-positive islets. But in the diabetic state, more than twice as many islets were located less than 1.6 micrometers from the innervation. So there was a strong shift in alpha cell nerve distance in the diabetic state. In the STZ model, the these associations were less clear. So again, control mice were very similar to C57 black control mice, which makes sense because they're the same. There was not much of an effect on insulin-positive islets and nerve distance in the diabetic state. And with glucagon, it was hard to draw any conclusions. There were more alpha cell clusters located in a close proximity to nerves. But it's hard to say, that it changed with the progression of diabetes. But when we looked at the human donors, there was a very strong shift again towards more insulin-positive islets located less than 1.6 micrometers from innervation, 11% in the controls and 29% in the diabetic state. And we use this distance because that's the type of resolution that we get from the light sheet microscope. But this tells us that there's some sort of altered nerve endocrine cell association going on in diabetes, and we wanted to investigate this more closely. So that led us to look at the endocrine nerve contacts in these samples. And we also wanted to look at intrapancreatic ganglia. As you can see in the video on the left, this is a 12x light sheet image. And here you can see that there are plenty of ganglia located really close to the islets, and the islets are also very densely innervated, as we already established before. For this data, we used 10x confocal imaging because we needed to be able to distinguish between individual cell bodies to be able to do these analyses. So first I go into a little bit more detail about analysis, beginning with the ganglia. So for this, we used Z-stack data to identify cell bodies, because it's actually really hard to identify cell bodies in 3D. So we used 2D data, and then we used images to create volumes around individual ganglia. And again, this is outlined more in detail in our bioprotocol that will be out any day soon. We calculated the number of ganglia per cubic millimeter. We calculated the average ganglion volume and the average distance between ganglia and insulin-positive islets. For the nerve contacts, we again used the distance transformation analysis as I talked about earlier, but this time we measured the distance between NF200 positive islets, NF200 positive innervation, and individual alpha and beta cells. And we used a distance of 0 to indicate a nerve contact. And that's illustrated in the images here. On the right, you can see the distance transformation analysis. And you can see individual beta cells with a color overlay defining how far away from the nerve they are. So beginning with the intrapancreatic ganglia in control mice. So here we didn't see any regional variation in the number, in the ganglion volume, or in the distance between NF200 positive ganglia in control mice when comparing the duodenal and the splenic region. When looking at the endocrine nerve contacts in control mice, we didn't see, again, any regional difference in the number of beta cells contacting nerves. When we looked at the number of beta cells contacting nerves in islets of different sizes, we saw that more larger islets had more beta cell nerve contacts. Large islets were defined as islets consisting of 100 or more beta cells, whereas small islets here were defined as islets consisting of 10 or fewer beta cells. But when we calculated the number of beta cell contacts as a percentage, then there was no difference based on size. In NOD mice, we found that the number of ganglia per cubic millimeter was not significantly changed, and there was no change in the volume of ganglia, but we did see that the distance between ganglia and insulin-positive islets was significantly prolonged in the diabetic state. And this is simply due to the fact that they had such a dramatic islet loss. As for beta cell contacts, we didn't see any significant change in the number of beta cells contacting nerves, and we didn't see any significant effect on the number of alpha cells contacting nerves. One thing that is very interesting to note, however, is that there are way more alpha cells than beta cells with nerve contacts. It's almost twice as many alpha cells than beta cells that are contacting nerves. In STZ mice, we didn't see a significant change in the number of ganglia, but we did see a significant loss of ganglion volume 15 days after STZ treatment. And this might be due to the overall toxicity of STZ, because unfortunately, STZ does not only attack beta cells, but we don't know what the reason for this loss in ganglionic volume is. Then there was no significant change in the distance between insulin-positive islets and NF200 positive ganglia. And again, because these mice had more, they didn't have a loss in the number of islets, but rather in the beta cell mass. So it actually makes sense. Looking at the beta cell contacts, again, no significant difference with the progression of diabetes and not in the number of alpha cell contacts. But again, we saw that in this model, there was significantly more alpha cell contacts versus beta cell contacts. In diabetic donors, we didn't see a significant loss in the number of ganglia, in the ganglion volume, or in the distance between ganglia and islets. And we didn't see any change in the number of beta cells contacting nerves. Again, these patients suffer from type 2 diabetes rather than type 1 diabetes. But all in all, it's interesting to note that there's no change in the contacts between endocrine cells and innovation. So that suggests that endocrine innovation, at least NF200 positive endocrine innovation, is preserved in the diabetic state. Then finally, we wanted to look more closely at sympathetic and parasympathetic innovation. And on the left here is sympathetic innovation. We stained for tyrosine hydroxylase, and this is an image acquired at 4x magnification. On the right, we have a pancreatic section stained for vesicular acetylcholine transporter, a marker of parasympathetic innovation. And as you can see, these two different branches of the pancreatic nervous system are really structurally different, with parasympathetic innovation, in this case, VSHT positive parasympathetic innovation, being incredibly dense, whereas the sympathetic innovation has more like these big branches. So in control mice, we didn't see any significant regional variation in exocrine and endocrine innovation that was positive for tyrosine hydroxylase. But we did see that in a way this mirrored the NF200 positive innovation, in that endocrine innovation was enriched compared to exocrine innovation in mouse islets. As for parasympathetic innovation, defined as VSHT positive innovation, again, there were no significant regional variations in exocrine or endocrine innovation, but again, we did see an enrichment of innovation in the endocrine pancreas versus the exocrine pancreas. And of course, it will be very interesting for the future to see, like, what do these markers, how are they affected in diabetes? What we could see from this data was that these markers work really well with our protocol. So that's holds promise for the future. But at this point, this is where our story ends, and I'm going to just go over the conclusions of this data briefly. So basically, we found that innovation is highly enriched in the mouse endocrine pancreas, with significant differences between the duodenal and the splenic regions. And we found an about 10-fold enrichment in the mouse endocrine pancreas when looking at NF200 as our pan-neuronal marker. We also found that there's a close association between nerves and islets in both mouse and human pancreata, which is maintained in diabetes. Islet innovation was enriched in the pancreas of diabetic NOD mice, and also during the progression of type 1 diabetes induced by STZ treatment. In both cases, we saw a twofold enrichment. We also found that islet innovation was enriched in pancreata of human donors with type 2 diabetes. Of course, for the future, it will be very important to determine the cause of this potential neuronal remodeling in diabetes. And of course, importantly, the consequences of this neuronal remodeling in diabetes. And very importantly, we need to study the individual contributions of the sympathetic, parasympathetic, and sensory innovation in diabetes. So with this, I just want to thank our colleagues at the Diabetes, Obesity and Metabolism Institute, at the Department of Neuroscience, and at the Microscopy Core and Advanced Bioimaging Center at Icahn School of Medicine at Mount Sinai. And with that, we are ready for questions. Thank you so much for listening.
Excellent. Well, thank you so much, Dr. Alvarsson and Dr. Stanley, for the really fantastic presentations. First question here, do you know if there's any role of reactive oxygen species in causing damage to the nerve covering, resulting in a loss of glucose control? And could we, protect the development of type 2 diabetes, for example, by eating foods rich in antioxidants?
I can try and answer that. So I don't know of any work that's been done looking at the effect of ROS on nerves in the pancreas at the moment. But I think that's a very good suggestion for a future direction for this work. There's definitely a lot of evidence to suggest that it plays a significant role in beta cell dysfunction.
Yeah, it's known that in neuropathy, reactive oxygen species are important. And I know that people suggest that eating antioxidants can be beneficial, but I think that studies actually trying that in patients have been quite disappointing. Unfortunately, like for many other diseases, but it's not known really in the pancreas.
Fantastic, yeah. Excellent. So next question here, are there any major circulatory changes in pancreatic innervation during hind limb unloading?
I don't know of any studies that's looked at that, actually. Alexandra?
Yeah, I have not seen any studies related to that, so I don't know.
Fantastic. Okay, so does glucose homeostasis decline in cognitive areas during normal aging, even without diabetes?
So as far as I recall, there are some studies that show that glucose uptake into cognitive regions, white matter, for example, does decline. There have been some functional studies using PET, which suggests that there's glucose hypometabolism as a consequence of normal aging. And I think there have also been some animal studies that have suggested that that occurs as well. I'll see if I can find a review that we can post into the resources to answer that question in more detail.
Excellent. Thanks so much, Sarah.
I've definitely seen studies on the hippocampus, where it's been studied in aging, and that there are differences. But again, we'll try to find some studies and get back regarding that. But yeah, there seems to be changes.
Perfect. Yeah, thanks so much. Next question here, is it possible that variation in the extracellular matrix in control versus diabetic mice and healthy versus diabetic individuals accounts for variation in the distance between certain cells and nerves?
Gosh, that's a really great question.
Yeah.
Again, I haven't seen anything. How about you, Alexandra, in the literature?
I haven't seen anything. I think it's a really interesting question, and I haven't seen anything on it, but I'll definitely read up on that because I think it's a good point. But I don't know of any studies looking into that.
Excellent. Another question here. Have you looked for innervation in somatostatin delta cells?
We have tried to stain for somatostatin, and we haven't looked specifically at innovation endocrine innovation touching these cells or nerve contacts, but it's something that we might be interested in doing in the future. We have an antibody that works really well for that, and we do, it's in our paper, and it will also be in our bioprotocol, but we haven't looked at it, unfortunately. But that would, of course, be very, very interesting and very important to look at.
Fantastic. Another question here. In many tissues, neurons have been shown to affect immune cell behavior. Has anybody looked if neurons in pancreas can affect the development of autoimmunity?
There was actually a very interesting paper which looked at sympathetic stimulation and sympathetic ablation and the effects on autoimmune diabetes. Again, I can post that into the resources, but there was evidence from that paper that suggested that if you blunt the sympathetic activity, then you may be able to attenuate the inflammatory response in diabetes. I think that was correct. But I will post that into the resources.
Yeah, it's a very interesting paper. Guyot et al. 2019. I think it was was it a Nature paper? So they basically electrically stimulate sympathetic nervous system in NOD mice, I think, and they don't develop insulitis. So that's very interesting.
Very cool. All right. So next question here. Do you know if the amount of innervation depends on the duration of the diabetes?
Yeah, so there are conflicting results on or findings on inflammation and how if that actually upregulates innervation. That has been seen with autoimmune disorders, for instance. And there is definitely evidence in the pancreas as well, in islets, I think from NOD mice, that it seems to upregulate. But there are also studies showing that sympathetic innervation, for instance, is downregulated, or it seems to depend what marker you are looking at. So there's a lot of conflicting evidence, but it seems like inflammation per se might be able to recruit innervation. Do you have anything to add, Sarah? There are many conflicting reports out there.
Yes, that's definitely true. I think there was one paper which suggested that there may be regional differences, so that there is possibly an increase in innervation close to the inflammatory markers, which is then lost further away.
Fantastic. All right, great answer. All right, next question here. Have you found any correlation between your diabetes model and pancreatic cancer? And if so, do you know if there's any pancreatic location similarities or differences?
We haven't looked. I think, again, that's an interesting question, but no, we haven't looked at pancreatic malignancies. I think there are groups in Pittsburgh who've examined innovation in pancreatic cancer. So they have some publications on that, but it's not something that we've looked at.
Excellent. Okay, next question here. Did you embed your pancreas tissue in agar blocks for the light sheet imaging, or how did you do that?
Yeah, so we used 1% low melting point agarose for that. Basically, you need to preserve the integrity. The pancreas is a floppy organ, so it's impossible to image it, unfortunately, to mount it if it's not embedded in agarose. And I use low melting point agarose because I'm worried about the temperature actually damaging it. So you want something that is fairly room temperature when you add the pancreas. So that's why I use that. It's slightly less hard than regular agarose, but it works fine for mounting it in the cradle for light sheet microscopy, and also for confocal imaging, where we just place it on a cover slip.
Fantastic. And I think in the interest of time, we'll just have one last question here. Do you have a sense of the VACHT, the staining phenotype, in non-diabetic human tissue?
Yeah, so I tried that. Oh.
Go ahead, Alexandra.
Yeah, sorry. I tried it in a very small section just to see if it would work. Actually, the same antibody that I used for mice, which I think is supposed to work in humans, but I couldn't actually, it didn't actually stain anything for me. again, unfortunately, these samples are so precious that I haven't been able to play around with antibodies. But yeah, in my hands, it did not work. That doesn't mean it didn't work. The issue that you have with human samples is, of course, that they are not profusely apostrophics, and the quality will be slightly less perfect. So that could also be something that interferes.
Sorry, but that'll just put you on mute for just a second. Sarah, do you have anything to add to that?
No, I was going to say the same thing, that there's a lot of variability in the quality of the human samples, but I think there may be work from Tang et al., which shows that there are some VACHT positive fibers within the human pancreas, but we haven't been able to look in our models from, our samples from people with type 2 diabetes or without.
Fantastic. Well, thanks so much, Sarah and Alexandra, for the really fantastic insights today. It's been a real pleasure having you with us.
Thank you, Liam.
Thank you so much for the invitation.
Yeah, and a big thanks to everyone also for joining us today to attend the webinar. And we'd also like to give a big thanks to Miltenyi Biotech for sponsoring the event. And so in closing, thanks again for taking part in this Inside Scientific webinar, produced with the American Physiological Society and Miltenyi Biotech. And we look forward to having you again next time.